Endomembrane trafficking is the flow of membrane material in the cell that helps in transporting proteins and other molecules to different locations in and outside the cell. What I will be helping you with today is having a better explanation of what goes on inside the cell. The cell is composed of a nucleus, which is the control center of the cell. In the nucleus, there is DNA and mRNA, which the mRNA will be sent out of the nuclear pores and into the cytoplasm, but we will be talking about that in a little bit. DNA is the blueprint of the cell that is composed of genetic codes such as adenine, thymine, guanine, and cytosine. The outside of a cell is called the plasma membrane. It is a biphospholipid layer that has hydrophilic heads and hydrophobic tails. Hydrophilic means water-loving and hydrophobic means water-fearing. Everything inside the plasma membrane and outside the nucleus is a cytoplasm or cell fluid. In the cytoplasm, there are organelles such as the endoplasmic reticulum, ribosomes, and Golgi apparatus. The ER has two distinct types. The rough ER has ribosomes attached to it, and then there's a smooth ER. The smooth ER synthesizes proteins and carbohydrates, while the rough ER modifies and packages the synthesized proteins throughout the cell. The function of ribosomes is protein synthesis, which is creating new proteins. The Golgi apparatus is the post office of the cell and gives a pancake-like appearance. The flattened chambers of the Golgi apparatus are called cisternae. At the beginning of the Golgi apparatus is the cis phase, which is more towards the endoplasmic reticulum, and that is where cisternae are being made. At the end, it is called the trans phase, where they secrete vesicles to other places in the cell. With protein synthesis, it involves transcription, which is reading copies of the DNA and making it into mRNA. The DNA goes into a DNA helicase, which will unzip the DNA into two strands. This will make the DNA copy become messenger RNA, or mRNA. In mRNA, there are individual copied triplets called codons, and these codons make up one amino acid. After transcription, there is a phase called translation. In translation, the mRNA strand leaves the nucleus through the nuclear pores and into the cytoplasm of the cell. The next thing that happens is the mRNA goes through the rough ER and attaches itself to a ribosome. Once the mRNA has attached itself to a ribosome, the tRNA comes and gives amino acids to the mRNA. The tRNA is transfer RNA. The transfer RNA has an anticodon that attaches to the mRNA codon. The anticodon is like a mirror image of the codon that needs to be paired together. When these two bond together, it starts a polypeptide chain. At stop codon, polypeptides separate. And that is what happens to the mRNA strand through translation. Another thing I will be talking about in endomembrane trafficking is post-translational modification, or polypeptide processing. This is when the mRNA goes through the nuclear pore and into the rough ER, or endoplasmic reticulum, where it makes proteins from the information given, and then it is put into transport vesicles and sent off to the Golgi apparatus. Through the Golgi apparatus, it will become synthesized and become mature proteins, which will either become a lysosome, which is a digestive enzyme that breaks down all cellular debris, or it will become a secretory vesicle that will leave the cell through a phase called exocytosis. Exocytosis is a process that the contents of a cell vacuole is released outside the cell membrane. And that is all that I will be talking about with endomembrane trafficking. Thank you.